So from the Hasselt University, we have Ann Kuipers here with the story of oxidative balance in plant stress responses. So losing or using.
Thank you very much. So it's always a balance between the prooxidants and the antioxidants. And you heard already a little bit from the previous one and also in the previous lectures in the past few days. And it is indeed about losing or using this balance in order to adapt to stressful environments. So if you lose the balance, then you get really stress as an organism, plants, animals, humans, doesn't matter. Or if you use the balance, then you can really use this prooxidants, let's say, to steer processes and really cope with the stressful environment. So it means that you always have to look actually at both sides. But how can you now investigate this oxidative balance? On one hand side, you can look at biomarkers of the oxidative balance, and then you have the real prooxidants and antioxidants. So the prooxidants, yeah, you can hear it. It's prooxidative, so it's really oxidizing. It's really not so beneficial. And the antioxidants, like it's mentioned here on this side, you can see a lot of prooxidants are free radicals. They are really not happy. And the antioxidants can really help them by giving them an electron and make them very happy again. So they can reduce them. On the other hand side, you can look at the outcome of the oxidative balance. And the outcome, the previous speaker already told so, it can lead to damage. It's more than the oxidative stress side, damage to different molecules, or it can lead to signaling. It's more the oxidative challenge side. So you steer processes directly via transcription factors or kinases. So the reactive oxygen species, for instance, the ROS, can directly activate a transcription factor or a kinase, and in that way start a signaling pathway, and hopefully you have some defense responses. And what they also do, they have a strong interplay with other metabolites or molecules that are important in signal transduction, like, for instance, calcium or phytohormones, or in our case, hormones. So the story I'm telling today is mainly about plants, but actually this scheme you can also use for animals, humans, and so forth. So first, let's have a look a bit closer to the biomarkers. So the prooxidants and the antioxidants. What exactly are they? And so the prooxidants or reactive oxygen species, I will say ROS because it's quite long to say the full name. What you see here in the second one, triplet oxygen, it's the normal molecular oxygen that we inhale. That's the normal one that is present in the atmosphere. And as you can see, it's a little bit a special one because it has two free electrons at both sides, but they have the same spin, so the electrons will not be shared between both atoms. So it's really a very special molecule because it's the only one in the ground state, so the energetic favorable state, that is like that. It means that the normal molecular oxygen is really not so reactive. It will not react unless you help it a little bit. And how can you help it to become reactive and then really reactive? It's on one hand side, you give it some energy so that one of the electrons, the spin flips to the other side, and you can share really nicely the electrons in this bonding between both atoms. And then we have singlet oxygen. So it's just energy. And I think we have been exposed to a lot of energy these days with a lot of sun. And the sun is the one who is giving a lot of energy with the UV, so that we really have a lot of singlet oxygen formation in our skin cells, for instance, our eyes. Those are very vulnerable in our case. On the other hand, you can also give electrons so that you really get it's a free radical, so one free radical. In this case, the hydrogen peroxide is not a radical, but it's also more reactive than oxygen, molecular oxygen. Again, an extra electron, and then you have the splitting in hydroxyl radicals, for instance, and they are also reactive. So you really can have this Ross projection via two important ways, or via energy, or via electrons. So these are some characteristics of those biomarkers. So we have here the different ones. So the superoxide, hydrogen peroxide, hydroxyl radical, all by the electrons donation, and then the singlet oxygen via the energy. What you can see is in seconds, the half-life, they have all a very short half-life. Some of the physiological concentrations, so really in the micromolar or even lower scales. The reactivity, and you can see they are all highly reactive except for hydrogen peroxide, which is mildly reactive, but it is because it's not a radical. And that means that it can also move quite far. Okay, 1 micrometer is not really far, but for a cell, it is already something. But if you want to measure in the end these Ross, then this half-life, which is really, really very short, is really problematic. It's really the challenge that you have to overcome. Okay, where are you now produced in plant cells? So we have the normal functioning. As we have the metabolic functioning also in our cells, for instance, here in the mitochondria, it's something that plants have, the respiration, just the cellular respiration. We also have it. You have at the end, you have the electron transport chain, and there, the word says itself, electron transport. If electrons leak onto oxygen, you have Ross. So they can be produced there. So that's an easy one. Where do we have an electron transport chain in plants as well? In the chloroplast, where we have the photosynthesis process. So here we have it as well. Okay, we also have other reactions there, but it's the same principle. You give electrons and you can have problems. You see here the lightning. That's the energy that comes from the sun. And so plants capture the energy from the sun and to start actually the entire process of photosynthesis. But energy from the sun means singlet oxygen formation. Then they have a separate pathway. So we have the photosynthesis that we all know, the respiration that we have and all know. But they have also another one, the photorespiration, and it's a bit an alternative in which 3 organelles work together, the chloroplast, the peroxisome, and the mitochondria. And also there you get a lot of production of these Ross. So this is the normal metabolic pathway. And if you have stress from outside, abiotic, pathogens, whatever, mechanical stress, you produce more at these metabolic levels because they will be disturbed, and as such, you produce more. And then you have also some enzymes, and a very important one is the RBOH, the respiratory burst oxidase, homolog in this case, because it's homolog to the one we have, for instance, in our macrophages. We call it the NADPH oxidases because they use NADPH or electrons from NADPH to reduce actually oxygen to superoxide. But it's outside the cells. So in the plants, it's outside the cells. They will be converted easily to hydrogen peroxide, which can easily travel inside. So these RBOH or NADPH oxidases, it's the same. They are very important also when it comes to stress. So normal metabolic function, and then these, they are important in development, but they are very important in stress conditions. Okay, if you then look to the other side of the balance, the antioxidants, in general, we can say we have 2 groups. We have enzymes that can detoxify these Ross, and you have metabolites that can do the job. If you look at the enzymes, and we saw already superoxide dismutase in the previous one, it's one of those enzymes. You can study it at different biological organization levels. The activity, really measure the activity in non-stressed and stressed samples by spectrophotometry, for instance, or you can check the protein abundance via Western blot or transcript levels using qPCR. So these are just different possibilities. Then you have the metabolites. Metabolites that can give and receive electrons, or that can capture energy, and in that way actually detoxify the ROS. How can you study them? By metabolite concentration, for instance, measure them, HPLC, spectrophotometry, whatever. Or you can measure the metabolites or enzymes in the biosynthesis or conversion pathway. So how they are formed and how they are converted to downstream products. Okay, the type of enzymes that we have the ones that directly detoxify the ROS. So in the first two cases here with the superoxide dismutase and the catalase, they only need a lot of the superoxide, and then you can see, okay, one is back oxidized to molecular oxygen, the other one is reduced to hydrogen peroxide. Catalase only needs hydrogen peroxide, will detoxify it by reducing it further to water or oxidizing it to molecular oxygen. And in that way, they don't need something else because they provide the electrons or interchange them between the molecules. But then here, this hydrogen peroxide can only also be detoxified by peroxidases, in this case, ascorbate peroxidase, for instance, to reduce it further to water. And then this enzyme also needs supporting enzymes and metabolites as electron donors. And these metabolites you can see here, for instance, glutathione, ascorbate, they can be oxidized to dehydroascorbate and glutathione disulfide, and they have always to be regenerated. So we call this supporting enzymes. And in the end, the final electron donor will be NADH, NADPH, or something like that. But what is the challenge of measuring then here the metabolites? You always have to think about the oxidation state, the redox state of these molecules. Some other metabolites, so here we have this hydrophilic ascorbate and glutathione, for instance. Same is for tocopherols, so like vitamin E, for instance. But carotenoids are really important in, for instance, capturing this light energy. Then the outcome, very briefly, I'm not going to talk about how we can measure that because it's the outcome, and I want to focus on the biomarkers and more specifically on the ROS. But it was nicely introduced already by the previous speaker that you have on the lipids, on the proteins, and on the DNA, a lot of damage that you can get. And I think on the DNA damage, you saw with the COMET assay and the DNA repair, many lectures that really introduce you into that. And of course, also the carbohydrates. If you then see to the oxidative signaling, so the positive outcome, and it's never one or the other. It's most of the time, it's a mixture of a bit of oxidative stress and oxidative challenge. So you always have those two. So if you have a stress factor, it can be abiotic pathogens, like for instance, metals. It's clear that they introduce oxidative stress or an oxidative disruption of the oxidative balance in organisms via metabolic disruptions. You can think on the respiration, or via the stimulation of these RBOH and the NADPH oxidases. But at a certain moment, you will have the production of reactive oxygen species in a certain amount, in a certain time. And what they will do, on one hand side, they will directly oxidize transcription factors that can be, that then activate a response, or they can oxidize the kinases, several kinases, also in our system, that then lead to a signaling response. As you can see here, they have a lot of interplay also with other phytohormones that are very important in signal transduction. But okay, how can we really investigate now from a methodological point of view this oxidative balance? It always depends on your research question. What do you want to know? Do you want to focus on this side, so the biomarkers, or preferably on the outcome? Most of the time, it's a combination of both. Your experimental design, in which species, in which setup do you want to look for it? What is the best one to approach, to use it, and then really the methodology that you're going to use. Oops, and I will work now, or I will present mainly now only the biomarkers related to the ROS itself. So I will focus on the steady state measurements, but it's important because you can use it throughout the research line of from fundamental to applied research. So that's really the important one. So we have the model organisms. Sophie already introduced Arabidopsis 2 days ago. So it's a model organism, very easy to work with, very small genome. You can easily transform it, grow it from seed to seed in 8 weeks. So wow. On the other hand, you have also crops, which are from an economic perspective, much more interesting, because that's really the ones that are going to do it. But then how you make the transfer from this side to that side, it really depends on your experimental setup. If you work in vitro, it's really strictly controlled. So you really know what you do. And for instance, in the case of the metal pollution, you add a metal and then you know, it's only the metal that is the difference between stressed and non-stressed. The further you go to the reality, so going to a hydroponics, to a pot system in the greenhouse, to a semi, mesocosm, let's say, to the field, then you go to the reality, but you cannot control all the parameters anymore. So that's then the disadvantage. So you really have to think, also if you work with animals, if you work with cells, or you go to real intact animals and animals in wildlife, as was done before, you really have to think, okay, what can be always the influential parameters of what I measure and see? Okay, but I will work on the ROS and I will focus only to the steady state analysis of these prooxidants, of the ROS. And there are actually two, there are more than these, but these are actually measurements that are very often used in research. So you can image where these ROS are formed after infiltration, and you use or chemical stains or fluorescent probes. Or on the other hand, you can really go for a quantitative analysis. Then you make an extract using again fluorescent probes or a spin trap technology. Okay, imaging after infiltration with chemical stains. Then you use a stain, in this case NBT, for instance. And as a chloride, NBT is soluble. And then if you add superoxide in this case, then it will convert to actually a formazan, which is an insoluble precipitate that you can see. It's a bit, it's saying here purple, but it's more bluish purple that you will see. Hydrogen peroxide, so the DAP staining that you can use. So this is the DAP, so this is the chemical stain. You use a peroxidase, harsh reddish peroxidase, hydrogen peroxide, of course, and you get again here an oxidation, and in the end, you get a polymerization that looks a bit brownish. So if we then look to some pictures, that's what you get. So if you add cadmium, so you destain, of course, your leaves. If you add cadmium, you can see, okay, the color is maybe not so good, but it's actually hardly any blue dots. Okay, the normal metabolism will always give you some ROS formation, but after cadmium, I think you can see in the wild type, so nothing special, that we have much more. And you can see it maybe, but you have to believe me, on the screen, on the television, it's much clearer to me. So hardly any under control conditions and much more brown when you expose them to cadmium. And then they made some transforming lines with an enzyme that is actually important in detoxification. But you can see here, different lines, different transformants, and actually the G6 is not really working because it's more or less comparable with the wild type. So if you then really want to continue, you really have to go for the other lines that are really more beneficial. Okay, these were the chemical stains. Then we go for the fluorescent probes. And an important one here is dihydroethidium. So it's this molecule, you add superoxide, it is converted, and you get actually your hydroxyethidium, which is fluorescent. So you can see it as a red color. On the other hand, what you can also add is, for instance, this mitosox red or mitotracker in our case, if you want to use animals. And so with mitosox, you can also add, it has a similar excitation. And what it gives, it can tell you then where the majority of your superoxide is formed. But there is one very important one, it's this one. You have also some non-specific oxidation. So you really have to be careful when interpreting the results. Okay, it's less than the superoxide, but you still have it. So you really have to optimize the protocol very well. And so here is an example. The control, you don't see anything. These are root tips. Then different treatments, aluminium, sodium chloride, cadmium, and copper. And you see that in some cases they produce more superoxide, but actually in all treatments you have more superoxide production. A similar fluorescent probe for hydrogen peroxide. So you have the, it's a very long name. You can read it here, but it's a diacetate in the end. When it enters the cells, some esterases will cut off the different acetyl groups at the end. It's still not fluorescent, but then if you have the ROS present in your cells, you get a fluorescent probe and you can see it. Okay, so if we compare those two kind of imaging with chemical stains or fluorescent probes, you can do it on intact plant organs and really see where it's produced. But we have to say for the chemical stains, it's more on the macroscopic level, while on the fluorescent probes, you can do it at the cellular level, even subcellular, if you add another stain for the mitochondria, for instance. The amount of ROS you can calculate, but it's semi-quantitative based on the amount of pixels or fluorescence intensity. The equipment, very easy, just a normal camera. Here you need already a fluorescence microscope or even a confocal if you want. What does it cost? Also important if you want to do research, it's cheap. This one is cheap. The chemical stains is cheap. The other one is more expensive. And some remarks. Of course, you have to destain if you want to use the chemical stains. And in the case of fluorescent probes, you also have to be careful about autofluorescence of your organism, but also the fluorescence photooxidative damaging of your probe itself. So it's a bit tricky, but okay, it works. It works nicely. If you then really want to quantify really exactly, then we have on one hand side fluorescent probes, on the other hand side spin trap technology. In this case, for hydrogen peroxide, we have amplex red. It cannot enter the cell as such as this molecule, but you add the peroxidase and you add hydrogen peroxide, then you get this resorifin. But okay, this is the reaction, but we have to get it into a cell. So if it's not cell permeable, what can we do to get it in interaction with the hydrogen peroxide, which is inside the cell, and the peroxidase we add? So how do we get it in contact with the hydrogen peroxide that is inside the cells? What do you think? What do we have to do? If it cannot cross the cell membrane, what do we have to do? How can we get the hydrogen peroxide in the cells, out of the cells, that they are coming free? What do you do? Yeah, you remove the cell membrane or you make an extract. That's actually what you do. So you crush this cell membrane so that you really get the access to the hydrogen peroxide. So we make an extract. And then I have the protocol here. So you really have to be careful because you remember, millisecond is the half-life of hydrogen peroxide. It means you have to work very, very cold. Therefore, the liquid nitrogen. Here, the liquid nitrogen. Then you have your extraction that you make. You add the amplex red working solution, so you add this without anything, so the negative background, so the sample background. Then you have the sample itself that you really want. You also do the same for a real dedicated hydrogen peroxide standard curve, and in that way you can start to make actually. You put it into the spectrofluorimeter. Often it is a microtiter plate, but It can be different things, and you measure the fluorescence in this case. So it's a fluorescence. So it means it's a lot of steps, but working code is really the message that you have to do here. And then you have the spin trap technology, and we have electron paramagnetic resonance or electron spin resonance. So it has always to do with this free electron and the spin plus a half or minus a half that it has in a certain magnetic field. And a free radical is always paramagnetic, so then you can detect it. If it is connected and it is diamagnetic, then you cannot, because you need a magnetic field, you cannot detect it. But as all our radicals, and it means superoxide, hydroxyl peroxide, they have a short half-life, you really need a spin trap to catch it, to really catch it so that it becomes more stable and you can put it into the machine and then measure it. So you have the radical, for instance, the superoxide or the hydroxyl radical. You have your spin trap. In this case, you see it has no free electron, so it's diamagnetic. And if you combine both, then you get the radical adaptive. So it's really combined, and this you can detect. It's a stable form, and you can detect with EPR spectroscopy. And these are typical kinds of first derivatives of the measurement, and you get these kind of patterns. But it's a very important technique to analyze unpaired electrons. So if we see which ones are detectable via EPR, you can see it here, superoxide, hydroxyl radical, and all kinds of radicals that are connected with some organic molecule, lipid radicals or something like that. Also for the nitrogen species, nitric oxide, for instance, you can measure, but not the other ones. So hydrogen peroxide, you cannot measure via this technique. This is an example of some research that we performed in the past. So yeah, this is very clear. No cadmium, 1 micromolar, 5 and 50. And they were really grown for a long time in cadmium, and you really can see what it does. So the purple color, it might be very nice, but it means that they are really investing in a pigment to help themselves to survive. But yeah, in the end, it will not work. But what you could really see here, you have the leaves and the roots, and at the bottom, you always have the simulation with a very specific chemical compounds here as well. This is a simulation for the superoxide. This is for the hydroxyl radical. And then you can see here, when you increase your concentration also here in leaves and roots, I hope you can see it, that at least the intensity of my peaks is becoming larger when I increase the cadmium, and it really resembles the pattern of the simulation. Here the same, you really can see that at 10 micromolar, it really increases. And this was done after 1 day of exposure. So it really goes very fast. If you then compare both techniques here, so they are quantitative, you need to make an extract. You can localize it, but how? It depends on what you sample. You can sample root tips or entire roots or leaves or whatever, or entire rosette. But you will lose the subcellular localization, of course, unless you harvest them separately, but that's really complicated to keep it then all stable. Absolute value is based on a standard curve or the peak height in case of EPR. The equipment, a spectropluorimeter or spectroscopy, EPR spectroscopy. The first one, fluorescent probes, it's expensive. With EPR, using the adapts and using the device, it's very specialized, so it's very expensive, it's very complicated. And then some remarks, and so the non-specific oxidation of your probe, the same as in the previous. And then in the last one, you can only measure radicals. You cannot measure, for instance, hydrogen peroxide. But if you want to know if something is happening with hydroxyl radical, it's only possible with spin trap, because we have no stain, no fluorescent probe that really is specific for that loss. So bringing them all together, it's an overview. You really have to think about the methodological approach. What do you want to know? And what is then the best option, taking all different steps and parameters into account. Yeah. Do you have this kind of device? No, we don't have it in our lab. So yes, I collaborated. So that's also important. I collaborated with people from Hungary to perform these experiments. So that's really, you have to see what is possible, what is not possible, and what is the best way, taking all parameters into consideration. So if we then look to the more steady state that are commonly used, they have some limitation, limited, not limited, spatiotemporal resolution. They are destructive. So if you want to know in time, you can do it partially in vivo, that's correct, but it's a steady state, so it ends. Or you have to harvest at different time points, which is quite labor intensive, let's say. And then it's often that some of these things are not really so specific. So that's a bit a disadvantage, but you always have your negative control and then your stressed plants, for instance. But what is a very big advantage of these techniques is that you can apply it universally for all plant species, no matter which ecotype, no matter whether it's a mutant or a transformant, you can do it. You always have to optimize the protocols because every plant is a bit different, but you can actually, in theory, use it universally. So that's a very big advantage of these type of measurements.